All right, today we're going to dive into a world that's totally invisible to the naked eye. It's a world where the instructions for life itself are written in a code of just 4 letters. We're going to explore the incredible tools that let us not just read that code, but copy it, make tons of it, and believe it or not, even rewrite it.
I mean, just think about that for a second. For pretty much all of human history, this blueprint of life was a total mystery. And now, right now, in our lifetime, we've cracked it. It's become a language that we're not just reading anymore, we're actively editing it with a kind of precision that used to be pure science fiction.
So, how did we get this incredible power? Well, it didn't just happen overnight. It's really the story of 3 groundbreaking technologies, and each one was a huge leap forward, taking us from just watching life happen to actually starting to shape its future.
So our story kicks off with a massive challenge for scientists. Imagine you find one single, super important sentence in a library with billions of books. Now, to really understand what it means, one copy is not going to cut it. You need millions. So how do you do that? Well, you invent a molecular photocopier. And that photocopier is called PCR, which stands for Polymerase Chain reaction. The idea behind it is actually beautiful in its simplicity. You take one teeny tiny specific piece of DNA, and in just a few hours, you make billions of perfect copies. It lets us take something completely invisible and make enough of it to actually see and work with.
The whole thing is this really elegant 3-step dance, and it's all controlled by changing the temperature. First, you crank up the heat to separate the 2 strands of DNA. Then, you cool it down just enough to let these tiny guide molecules, called primers, find and grab onto the exact spot you want to copy. And finally, this amazing little protein, an enzyme, gets to work building brand new DNA strands. Then you just rinse and repeat, and the copying begins.
And this is where it gets crazy. This is where the exponential power just takes over. Each cycle doubles the amount of DNA. So 1 copy becomes 2, 2 become 4, 4 become 8. You get the idea. Within just a couple of hours, you've gone from one single strand to literally billions of identical copies. It is a full-on amplification explosion.
Now, being able to amplify DNA like this, well, it changed everything. All of a sudden, we had this incredibly powerful tool for all sorts of things. You know those COVID tests we all got so familiar with? That's PCR. It's what's used at crime scenes to identify a suspect from a single drop of blood. It helps us trace our own evolutionary family tree. It basically gave us the power to read the book of life in amazing detail.
But you know, for all its power, PCR has one big limitation. It's kind of a lab rat. It needs a special, expensive machine to do all those temperature cycles. So what happens when you need to diagnose a disease in a remote village, miles and miles from any lab? Well, that's where our next technology comes into the picture.
And this visual just says it all, right? On one side, you have the high-tech PCR machine, totally stuck in a lab. On the other, you've got a scientist out in the field with a simple, portable device. This was all made possible by a technology called LAMP, which was cleverly designed to work at one single constant temperature, making it completely portable. LAMP stands for Loop-mediated Isothermal Amplification. And the key word you really got to remember there is isothermal. It just means one temperature. No complicated machine, just a simple little heater. For diagnostics, this was an absolute game changer. So how on earth does it copy DNA without constantly changing the temperature? Well, the answer is a brilliant piece of molecular engineering. Instead of using heat to separate the strands, LAMP uses a special enzyme and a clever set of primers that actually cause the DNA to fold back on itself, creating these little loop structures. These loops then become brand new starting points, kicking off this super fast, non-stop chain reaction of copying. It's genius.
So what does this get you? Well, you end up with a test that is incredibly fast. I mean, we're talking results in under an hour. And it's so simple that a lot of the time you don't even need a special reader. A positive result can just be seen with a simple color change in the tube. This technology literally took the power of molecular diagnostics out of the lab and put it into the hands of people, right there in the field.
Okay, so we can copy DNA with PCR, and we can detect it anywhere with LAMP. But this final leap in our story, it's the most profound one of all. We are now moving beyond just reading the code to actually rewriting it.
And this is a fundamental shift. I want you to think of PCR and LAMP as being like detectives. They're great at finding a specific piece of DNA and telling you, yep, it's here. But CRISPR is an editor. It can go into the code and actually change it. This is a level of control that we have just never had before.
And the craziest part?
This revolutionary technology wasn't invented in some high-tech lab. It was discovered in nature. CRISPR is the ancient immune system of bacteria. Yeah, for billions of years, bacteria have been using it to find and chop up the DNA of invading viruses. Scientists, in just a stroke of absolute genius, realized they could harness this natural system and turn it into a programmable gene editing tool for us.
Honestly, the easiest way to get your head around CRISPR is to think of it like the search and replace function in a word processor. It's got two main parts, something called a guide RNA, which is like the search bar. It homes in on the exact genetic sequence you want to change. And then there's a Cas enzyme, which is basically a pair of molecular scissors that does the cutting.
So here's how it all goes down in 3 steps. First, that guide RNA finds its target and locks on tight. Second, the Cas enzyme comes in and makes a super precise cut, breaking the DNA. And here's the really clever part. CRISPR then just steps back and lets the cell's own natural repair crew take over. The cell can either just patch up the break, which usually disables the gene, or, if we give it a new piece of DNA as a template, the cell will use that to make the repair, effectively pasting in our new, edited code.
The potential applications here are they're mind-boggling. In agriculture, we're already using it to engineer crops that are more nutritious or resistant to diseases. But what's really fascinating is that because CRISPR is so incredibly good at finding specific DNA sequences, it can also be used as a hypersensitive diagnostic tool, creating tests that can find tiny, tiny traces of a virus or a cancer gene.
So let's just pause and think about the journey we've been on. In just a few decades, we've gone from the really hard work of copying DNA in a lab to instantly detecting it anywhere in the world to now having the power to literally edit the code of life itself.
And you can really see the evolution when you look at them side by side like this. You start with the lab-based PCR. Then you get the fast and portable LAMP. And finally, you have the programmable power of CRISPR, which can both edit and detect. Each one built on the last, and each one opened up a whole new world of possibilities.
And that right there, that's the real story. This isn't just about making better lab equipment. This is a fundamental shift in our entire relationship with biology. We are no longer just observers of the natural world. We're becoming its architects.
Which leaves us with a pretty profound question, doesn't it? For the first time in the history of life on this planet, one species can consciously edit its own genetic code and the code of every other living thing. We've moved way beyond the question of can we to the much, much harder questions of should we and who gets to decide. The power to rewrite life is now in our hands. What will we choose to create?